into an examination of the structure and function of BHD enzymes, which I'm excited to give you a little bit of details about today. So as might have been evident from some of the publications that Lars highlighted, I came into this through my interest in how plants respond to their environment. And the way that I started was looking at plant light signaling. And I think light signaling is a really charismatic example of how plants don't have control over their environments, and they have to find a way of making do with what they have while staying put in the spot that they cannot move from. So this is a garden plot that I had back when I was living in New York, and I was a very ambitious first-time gardener. I planted my plants way too close together, and what happened was it created some really unideal light situations for these plants. So you can see that I planted them so close that they were crowding and shading each other. But plants are clever, and they're able to do things such as phototropism and bend away from the shade in order to optimize their light conditions. The same is true of different pests such as insects. The plants just can't shake off their insects and move on. And this is the same terribly blighted garden that I had that year. So we had lots of flea beetles that were predating on the tomato leaves. And we also had a fair bit of aphids as well. Tomatoes are an interesting example because they make acyl sugars. And it's not just in cultivated tomato, they're actually found throughout the Solanaceae family. Acyl sugars are these very sticky compounds that are produced by these plants. And they consist of a sugar core here, which is sucrose, with several esterified acyl chains to them. And you can see here in the case of wild tomato that it creates such a sticky surface on the leaves and stems. You can kind of see the sheen of the acyl sugars being exuded from the leaves on these plants, that insects just stick right to them. They're a great way that these plants can defend themselves against predators. And it's not just tomato and tomato relatives that have this ability. We also have different Solanaceae species, such as tobacco here. When you have a tobacco plant that is not able to accumulate acyl sugars, you can see that the number of whiteflies and aphids that survive on those plants without the acyl sugars is much higher than normal plants that accumulate acyl sugars normally. So this arsenal is really important for plant health and survival in situations where insects are predating them. So we don't have just acyl sucroses, although that's what I was just highlighting on the previous slide. You also can have different sugar cores on these acyl sugars. So in this case, you have one from Solanum pedoense that has an inositol sugar core, and the one from Solanum nigrum that has a glucose sugar core. And there's another layer of diversity when you investigate the acyl chains. So here I've colored them by the number of carbons in the acyl chain, and you can see that some of them are branched, some of the chains are straight, and we even have some evidence that I haven't shown here, but from other species that can even have hydroxylated acyl chains that can then go on and have further modifications. So even within these simple components, you end up with a surprising amount of diversity. And to describe these acyl sugars, we use this nomenclature that I'm going to be using a little bit throughout the talk. So the first letter describes the sugar core. So in this case, for this acyl sugar, we have sucrose with 4 acylations, 17 total carbons in the acyl chains, and 4 individual acyl chains, and the lengths are indicated. So how are these acyl sugars actually made? They're made by a large family of acyltransferases that are pretty ubiquitous in flowering plants called BHDs. And BHDs typically use an acyl acceptor, in this case it is sucrose, and an acyl-CoA donor. And the BHD enzyme, in this case ASAT1, transfers the acyl chain from the acyl-CoA to the sucrose, in this case to make a monoacyl sugar. And if you look at the cultivated tomato pathway, you can see that there's a whole team of ASATs that work together, where one ASAT adds one acyl chain to the sugar core to create the final acyl sugar that we observe. And if you look at the total number of acyl sugars that we can observe in cultivated tomato leaves, you can see that this is the most abundant one, this 2555. But there are several others, so they're not just extremely short chain. You could get some C10 and C12 length acyl chains as well. This pathway has been really well described and really well studied. So but we were absolutely gobsmacked to find that we missed some of the acyl sugars in cultivated tomato. So it's not just the trichome acyl sugars. In fact, the Aroney lab discovered that there are also acyl sugars in tomato roots. And so Rachel has gone ahead and annotated the root acyl sugars that are present in tomato roots. And you can see that there's a surprising amount of diversity of these root acyl sugars. There's a lot more of them than what we see in the trichomes. And you also see that they have more acyl chains than you see in the trichome acyl sugars. So you get up to 5 acylations, whereas in the trichomes, you get up to 4. But the biggest difference between these acyl sugars that we found once we started doing NMR with these is that they have a different sugar core. So as I've been describing, the trichome acyl sugars in cultivated tomato are sucrose. Sucrose is a disaccharide. It's composed of a glucose and fructose subcomponents, whereas what we see in the roots is a very unusual myo-inositol glucose core, where it is a disaccharide, but it's composed of glucose and myo-inositol instead. And just to emphasize the difference between these acyl sugars that we find in these different tissues of the same plant is that you have a different sugar core with more acyl chains and different acyl chain identity between them. So we surmise, based on the level of difference between these two acyl sugars, that they're probably not being made by the same pathway. It would be difficult for the substrate specificity of these enzymes to make both of these acyl sugars from these different components. So Rachel did some RNA-seq analysis to try to figure out which are the enzymes that are responsible for creating these root acyl sugars. So as I described, we know that ASAT1 from tomato trichomes catalyzes the first acylation on acyl sucrose. But what is the first acylation, for example, in tomato roots? Well, in the RNA-seq analysis that Rachel did, she found that there is ASAT1-like, which is a close paralog of the trichome ASAT1, and that it is expressed specifically in tomato roots, whereas the trichome ASAT1 is expressed specifically in trichomes. When she investigated some knockout mutants, she found that the trichome ASAT1, when you knock it out, predictably you get no acyl sugar accumulation in the trichomes. But knocking out that ASAT1 has no effect on the acyl sugar content in the roots. The opposite is true for the ASAT1-like enzyme that's expressed in roots. Like, it has no bearing on the trichome pathway, but when you knock it out, you no longer see acyl sugars in tomato roots. Based on this evidence, we thought that, yeah, ASAT1-like must be the first step in the pathway. So as the resident biochemist in the last lab, it fell to me to start characterizing this enzyme and trying to figure out in vitro, what is it actually catalyzing that contributes to this root acyl sugar pathway. So our kind of naive assumption was that the enzyme would use this myo-inositol glucose sugar core as an acyl acceptor, and maybe NC7-CoA as an acyl donor. It's one of the most common acyl chains that we see in the roots. When we purified the sugar core, because unfortunately one cannot buy this from Sigma, when we characterized this sugar core from purified acyl sugars, what we actually found, much to our disappointment, that it is not stable in water. So I wasn't able to get a lot of data using the sugar core because it is so labile, but my preliminary results suggested that ASAT1-like was unable to acylate this core. But to try to pick apart this further, we used sucrose as a representative disaccharide to see if ASAT1-like could acylate sucrose. And we also looked at glucose and myo-inositol, which are the individual constituents of that root acyl sugar. And what we found that was a little bit surprising was that ASAT1-like has really strong, well, fairly strong activity with inositol. It is able to acylate it without too much trouble, but it really doesn't have any activity with a disaccharide such as sucrose. And this was really interesting to us, but it presents kind of a problem because the root acyl sugars, as Rachel annotated them, have this inositol glucose sugar core. Starting the pathway with inositol kind of creates some confusion about how we go from inositol to inositol glucoses in the final root acyl sugars. But when you look at the fragmentation data, for example, for this triacyl inositol glucose that Rachel annotated, what we see is that 3 acyl chains are actually on the inositol core with none on the glucose, which leads us to the hypothesis that ASAT1-like actually does start by creating a monoacyl inositol from just the monosaccharide substrate. and that two further acylations occur, followed by a glycosyltransferase that tacks on this glucose to a potential triacylinositol. I'm actually going to move on now in the talk to another investigation into ASAT1-like activity, but I do want to say that we have some pretty good hypotheses about the enzymes that are involved in this bit of the pathway, and it really does seem that this triacylinositol is the intermediate on the way to making these final acyl sugars. So to conclude this part of the talk, interestingly and surprisingly, tomatoes make acyl sugars not just in the trichomes as has been thoroughly described in the literature. It also makes root acyl sugars. These root acyl sugars seem to be made by paralogs of the trichome pathway that are expressed specifically in the roots. And these paralogs have activity with inositol in some cases to create this eventually to create this unique inositol glucose core that hasn't been thoroughly described in Solanaceae acyl sugars quite yet. It also suggests that there's a acyltransferase in the pathway. So in terms of the acyl sugars that we have worked out the pathway before, this is the first time that we've had a pathway that's not just ASAT. It's not like just like this ASAT adds this acyl chain, that ASAT adds that acyl chain. There's another enzyme family, probably a UGT transferase, that is also involved in making these final structures that we observe. So I came into this through acyl sugars, but VHDs are really wonderful enzymes, and they're capable of catalyzing a really diverse set of chemistries. So you have these simple acyl sugars with sucrose and acyl chains, but VHDs are also responsible for the synthesis in plants of a lot of different molecules. So you've got cocaine, where an aromatic group is added to an alkaloid. You have terpenes like forskolin that get modified by VHDs. This is a cyclic AMP stimulator, which is, I think, in clinical trials for in the US. And you also have anthocyanins, which get modified by VHDs as well. In this case, this is the malonylation of the anthocyanin core. And what strikes me here is not just that you have like so many different chemistries and stuff, but so many of these molecules are really important, not just for plant survival, like acyl sugars or anthocyanin, which are really important in plant physiology, but you also have chemicals or metabolites that are important for humans. Cocaine can be used as an analgesic, and forskolin as well as treating certain diseases. So these enzymes are really interesting to study because there's like a large payoff in investigating their activity and what they can do. But one of the limitations of these is that they can, as everybody knows who works with specialized metabolites, is that they're confined to certain tissues. Forskolin is a really good example of this. It comes from coleus roots, and it's really hard to get decent amount of forskolin from the roots. And one approach to handling that is to use the enzymes that synthesize these compounds and make the compound in vitro. But this is also a difficult approach. And one of the reasons for that is that natural enzymes can be very slow, especially when they come from specialized metabolism. So this red line and this green line both describe the catalytic rates, the kcat of enzymes involved in primary metabolism. So you can see that they are catalyzing a decent amount of molecules per second, but enzymes that are involved in secondary, or as we say, specialized metabolism with this blue line, are significantly slower. And these slow rates can really slow down our ability to efficiently make some target compounds in vitro and adapt these compounds for better use for humans. So back to ASAT1-like. One of the reasons that it's interesting to use ASATs as a model is because they have really simple components. You've got myo-inositol, you've got NC7-CoA, which I buy, but you can also synthesize very easily in a day in your lab. And it makes this also pretty simple monoacyl-inositol structure that's easy to study and work with. So how is this catalysis actually happen? So if you look at a space filling model of a BHD, so in this case, this isn't the malonyl transferase of anthocyanin. I like this structure because it has a ligand bound to it. But when you look at the space filling model, what you see is that BHDs have a central channel on the inside. And this is where the molecules will go into the protein for catalysis. So if you look at the ribbon model of this BHD, you can see that on one side of this channel, you have the acyl-CoA donor is bound here. And then you have the acyl acceptor, which would bind in the other side, but is not in this crystal structure. For the transfer of the acyl chain from the acyl-CoA, you have a catalytic histidine, which in this case you can see is pointed right at the division between the acyl chain, which in this case is the malonyl group, and the CoA moiety. And this is basically how it happens. It's like a nucleophilic attack that allows this acyl group to be transferred to the acyl acceptor. So in my pathway discovery project that I was talking to you about in the first half of the talk, I was trying to characterize the cultivated tomato ASAT1-like to see how it falls in the pathway. And as a part of that work, I also looked at the activity of the wild tomato version of ASAT1-like. I was very surprised to see the difference in magnitude of activity between these two enzymes. You can see in this endpoint assay that the quantity of this monoacyl inositol that the wild tomato enzyme is able to make is just much larger than what we see for cultivated tomato. And that was pretty striking to me. When I followed this up further by looking at the kinetics, it further clarified this. So I've started using a new to me method for doing these Michaelis-Menten kinetics to measure catalytic efficiency in these enzymes. So I've showed you a few times this reaction mechanism. But what I haven't emphasized is that when this acyl chain is transferred to the sugar, it also creates free coenzyme A. Well, there's a reagent called DTNB that will react with free sulfohydryl groups like the one on coenzyme A and create a yellow color. So when you do this reaction in vitro, you get a very nice yellow color, and then you can actually get kinetics as well when you use a plate reader, and you can see how with different amounts of NC7-CoA, the velocity of the reaction increases in this classic Michaelis-Menten fashion. Yeah, and if you're interested in this and you're working on BHDs, I highly recommend that you check out this Sullivan 2023 paper in Methods in Enzymology. It's been a really big change in my work, and you don't have to use mass spec to do this. So it's been a great help for me. So when you look at the kinetic parameters of the wild tomato enzyme versus the cultivated one, what you see is that the wild tomato enzyme just clobbers the cultivated tomato enzyme in terms of its parameters. So if you look at the enzyme efficiency, you can see that with NC7-CoA, the wild tomato enzyme is 32 times better and more than 100 times better with the myoinositol substrate. So you have a wild enzyme that has like just really robust parameters and a cultivated one that is just really not good. This gets even more interesting when you consider the fact that these proteins are almost identical to each other. There's only 17 amino acids that differ between these proteins. And I surmised based on the fact that they're very similar, but there is a huge difference in activity, that one or several of these amino acid changes must be responsible for the difference in activity between these two enzymes. So I initiated a residue swap study. So the first thing that I did was I just grouped the amino acids based on their position in the primary sequence. So you see this group 2 set of residues here. You have 3 amino acid changes that are kind of on top of each other. So when I did my residue swap, I changed all 3 of these residues from their identity in the cultivated tomato enzyme to make them the wild tomato enzyme version. And that reduced my mutations to study from 17 to 10, made it a little bit easier. And out of those 10 groups of mutations that I studied by this residue swap, 4 of them ended up having a pretty substantial increase in the activity of the cultivated tomato enzyme. And when you overlay those 4 individual amino acid changes, or 4 groups of amino acid changes on this model of ASAT1-like structure, you can see that all of those changes model as facing the catalytic channel that I was describing earlier. And you can see that this one in particular, like this group 1 mutation, is right on top of the catalytic histidine. And this makes sense. The ones that are more distal or on the outside of the protein don't seem to have the same effect on catalytic activity. So when I tested these mutations and their effect on ASAT1-like activity in vitro, I saw that these individual mutations, as I kind of previewed in the last slide, are able to substantially increase the activity of ASAT1-like. It didn't really rescue the activity to the same extent of the wild tomato enzyme, but when I took these groups of mutations and then combined them, for example, to make a 1+4 mutant or a 1+9 mutant, what I see is that those combinations are able to restore the activity to the level of what you see in the wild tomato enzyme. that means that amino acid changes of just 1 to 3 changes is able to completely rescue the activity of the cultivated tomato enzyme and give it kind of the more robust features that we see in the wild tomato enzyme. So I was happy about this. I was quite excited to see that we were able to pinpoint some of these differences that lead to the activity changes. But I'm really interested in the mechanism. Can we take some of this information and can we apply it to PhDs so we understand how they work and not just ASATs or not just ASAT1-like? And for that, I decided to focus on the group 9 mutation. So it's not the shiniest mutation in terms of magnitude of activity increase, although it's one of the better ones. The reason that it really interested me is that there's a surprising level of conservation of this residue in BHDs. So in ASAT1-like, the cultivated tomato enzyme with less activity, it is a threonine. But if you kind of scan the clade 3 BHDs listed here, and it's not just ASATs, it's really other ones too, what you see is that the most common residue there is actually a methionine. And where it's not a methionine, it's quite often another hydrophobic residue. So this threonine, I predicted, would have to be very disruptive to activity. And where this threonine/methionine residue models in the structure of ASAT1-like is right next to where the acyl-CoA donor would dock inside of the protein. So when you compare this structure to the anthocyanin acyltransferase that I highlighted earlier, the residue would be about right here. Unfortunately, because this anthocyanin acyltransferase is not in the same clade as the ASAT1-likes, or it's not in clade 3 of BHDs, that residue is not a methionine. But since the structures are similar enough, when you overlay them, you can see that this residue ought to kind of be in the neighborhood of where that acyl-CoA would be binding. So I hypothesized that having a threonine here in the wild type cultivated tomato enzyme has a large reason to do with why the activity is not so good. But I decided to move away from ASAT1-like for further analysis. The reason for this is that ASAT1-like is not really that well expressed from E. coli. It's actually a fairly difficult enzyme to work with in vitro. But another acyl sugar, acyltransferase, ASAT3-like 5, has the conserved methionine at the position where ASAT1-like is a threonine. So I decided to work with this one because it expresses really well, and I can do kind of more detailed biophysical characterization with this nice stable protein. So the activity of this ASAT3-like 5, actually, I predict that it is in the root acyl sugar pathway and modifies the acyl inositols that I was describing earlier. So the hypothesized function for it is that it takes an NC7-CoA and acylates a diacyl inositol to create a triacyl inositol. So when you look at the wild type enzyme, it has robust activity for making this I319 product. But when that methionine has been mutated to a threonine to make it like that poor activity cultivated tomato enzyme, you can see that this enzyme also loses its activity when that methionine has gone away. And so now we have the case where we have two enzymes where we have, and they're really not that closely related. They're both ASATs, but they are pretty distal to one another. But you have two ASATs that are not very similar, where mutating this methionine in some ways has a really large impact on catalysis. So to follow this up, like I said, my hypothesis was that NC7-CoA binding inside of these enzymes is maybe a mechanism for this mutation. So to follow this up, I took this ASAT3-like 5 enzyme and then I put it in a calorimeter, and then the calorimeter injects doses of NC7-CoA into the protein. When the ligand binds the protein, in this case, heat is released. And when you track the release of heat over time as the ligand binds to the protein, you can actually quantify the association between the ligand and the protein, which again, I just could never have done with ASAT1-like because it's just, you can't get enough protein to do this kind of analysis. When I investigated the M to T mutation that had no catalytic activity or no enzymatic activity, what I saw was actually the association between the protein and the ligand was basically the same between these two things. So whatever is happening, it has a really strong effect on the ability to make the product, but the mechanism is not through acyl-CoA binding as I originally suspected. Unfortunately, for now, I don't have better answers about the mechanism for how this works. It is a bit of a mystery to me. But one thing that I would like to highlight is when I looked at these group 10 mutations, you can see that these two residues model as quite close to this threonine/methionine position, and they're quite distal to the catalytic histidine. So whatever is happening, there's a region of this protein that is quite far from where catalysis happens, but seems to be very important for activity. And some of these residues are also conserved, especially this methionine in clade 3 BHDs. So I'm currently trying to follow this up and see how they affect the activity of other BHDs and try to understand how is it that this section of the protein is having such a large effect when it's actually not so close to the catalytic histidine and is also not affecting the acyl-CoA binding in that region. So just to wrap up this section of my talk, rationally swapping amino acids between an efficient enzyme like the wild tomato enzyme and a less efficient one like the cultivated tomato enzyme was able to increase the activity of the cultivated enzyme by quite a lot. Out of those residues that I found that I swapped, this group 9 methionine that I found is conserved in a lot of clade 3 BHDs, and therefore this finding is likely to be applicable to other BHDs, not just to our little ASAT system. However, whatever the mechanism that this methionine/threonine is having on the activity of these enzymes, it doesn't appear to be through acyl-CoA binding, or at least not the actual binding to the protein. It could have an effect somehow during the catalysis. So I'm currently trying to follow this up and think about what it means and how we can apply this to BHDs more widely. Because like I said, the acyl sugars are really interesting compounds, but it would be great to take some of this information and apply it to like a forskolin acyltransferase or something that is really limiting the production of high value specialized metabolism compounds in vitro. So I want to thank everybody in the last lab for facilitating my work and having interesting conversations with me about this and really enriching my time here. I particularly want to thank Rachel. We collaborated really closely on the beginning of the talk on the ASAT1-like. She characterized all of the root acyl sugars, did the RNA-seq work that helped kind of find ASAT1-like to be worked with, and she was a really important part of that. Paul did the NMR structure of acyl sugars, or the root acyl sugars, that enabled us again to further understand what was going on. And I want to thank in particular Rhiannon, who is a very talented undergrad that I worked with, who helped me a lot in particular with those kind of double mutants of ASAT1-like. And thank you for listening.
Thank you very much, Jaynee. Great presentation.
Thanks.
For questions, just feel free to raise your hand and then, or just start talking.
I have a question if nobody else is asking. Can you hear me?
Yes.
So any clues on what might have happened during tomato domestication that made this enzyme, or that made the selection for this variant be predominant?
Yeah, we are toying with the idea that there was something to do with cultivation that kind of caused the cultivated ASAT1-like to kind of be gupped. I will say that there are 2 alleles of ASAT1-like, even in cultivated tomato, which might help support this. And they do have different degrees of functionality. So the ASAT1-like from Heinz seems like it's actually slightly better than like the M82, which is what we use as our model. So yeah, I think that there is some evidence that cultivation has had an effect on the root acyl sugars.
Cultivation or domestication?
Sorry, yes. Domestication, yes.
So just a follow-up, and again, for my own instruction, have the genes associated with domestication of tomato been mapped and characterized?
This is out of my wheelhouse. I'm not sure. I imagine so, given the level of importance of cultivated tomato to agriculture.
Okay. Thank you.
Yeah.
I have a question. This doesn't have anything strictly to do with the data that you presented, but I saw it in the news the other day, and I don't know, I find it fascinating. So there's been some talk in the news about artificial intelligence models that and how they relate to protein sequences, and Microsoft published one, I think, the other day. And
Yeah.
this was what was in the news, and it's for, studying, well, you can use it to do a variety of things. But I guess I was reminded of this article during part of your presentation, because you're talking about how, generally speaking, it looks like enzymes involved in specialized metabolism are slower catalysts. And usually I think about this as potentially being because they are, I think we've seen there's some evolutionary pressure to have some specialized metabolic enzymes that are kind of promiscuous and can do various things. And I feel like, and I've heard some people say that the promiscuity and the kind of catalytic efficiency are a little bit mutually exclusive.
Yeah.
I'm wondering if you've read anything or have heard anything about using some of these new tools, the AI tools or something similar to it, to begin to understand how to move along this spectrum from promiscuous to efficient. And you kind of, like, in your data, you have some kind of things that are related to that. Anyway, I don't know. I'm sorry if it's a silly question, but I find these things fascinating, and I'm always curious to talk to people that might know more about it.
Yeah, I don't think it's a silly question at all. I think that's very much where we ought to be moving. And you are correct. There is a trade-off between efficiency and like the number of substrates these enzymes seem to be able to manage. This paper that I got this kind of difference in catalytic or speed from really also emphasized that it might not be in the interest of the plants to have efficient specialized metabolism enzymes. Like, for example, the acyl sugars are actually a great example because they're sequestering sucrose and inositol, like these important energy sources, away from the plant. Like, you don't necessarily want good enzymes to be taking away from primary metabolism. And to Eric's point, like, this could be related somehow to the domestication of tomato. Like, maybe tomato fruits don't taste good when sugars are being used in the roots like this. That is wild speculation. I don't know. To your point about how AI could change this, I think that's really interesting. I mean, like, these kinds of things have already changed this, because I have like an AlphaFold model of ASAT1 like that is incredibly useful. And I didn't have one before the paper was released. So it's already been an improvement, and I'm excited to see how that goes and how it's going to inform like my work in this kind of effort.
Thank you.
Yeah, also feel free to post questions in the chat if you then we can ask this question, no problem.
I had a question. I don't know if everybody can hear me.
Yes.
That was super fascinating what you were just talking about, the specialized metabolic enzymes being sort of slow to go. And I also, this is totally, I hadn't heard this before, and I just want to learn more about it. Have there been expression studies? Because that's the other piece of the puzzle, is just to kind of turn them on late in development. It seems like those two could kind of talk to each other. Have people looked at that?
Yeah, it's a well understood principle of specialized metabolites in particular, that their expression is extremely confined to particular tissues. And it is a part of it, because a lot of these compounds, not acyl sugars necessarily, but a lot of them are toxic to the plants. And if you express them in leaf tissue, for example, in palisade mesophyll instead of a trichome, you really run into some issues. Yes, specific expression is an important part of that.
I mean, it's certainly true for the anthocyanin pathway, but that there's things that come on late. But it's so interesting that it's there for the acyl sugars. The other thing I was wondering about is I'm always thinking about compounds being sneakily expressed in other tissues. Because it's like what are the anthocyanin genes doing in spinach or something like that? I just think like maybe these compounds are being sneakily expressed, and I wondered, for the acyl sugars, do you think there's kind of other places or developmental times where they might be hanging out that might be other sort of parallel pathway things like what you found in the roots?
Yeah, it's a great point. I mean, just the fact that we belabored under the assumption that like acyl sugars were for trichomes for such a long time, to the extent that we, I think, kind of stopped surveying other tissues, is a really important example of how we've kind of specialized into a certain tissue type to study. But in fact, I think we are missing a significant amount of diversity. And that's one of the things that we're trying to do in the lab right now is to see like what other species are making root acyl sugars, and are there potentially acyl sugars in other places that we didn't expect? Yeah.
Cool. Thank you so much.
Thanks.
I also had one question. I was wondering, how much do you know, or how much is known about the ecological significance of these root acyl sugars?
The paper that originally characterized the root acyl sugars, or identified that they were a thing in tomato roots, found that they were induced by certain strains of like bacillus. So they were really doing like more ecological approach to understanding just exudation in particular. And they happened to find that acyl sugars were exuded in response to treatment with certain microorganisms in like hydroponics. So there's a fair chance that the role of the root acyl sugars is either to somehow recruit and prune the associated microflora with the roots, or perhaps as a defense, the same way that they are in the trichomes. It's hard to know right now. And it's also hard to think about how much we can draw from the chemical properties of the acyl sucroses, for example, which are really sticky on the leaves. Like, are the inositol glucoses, are they also super sticky in the roots? Like, are they providing a physical barrier? I don't know. There's a lot of questions there that should be addressed.
Thank you.
Mm-hmm.
Probably really difficult to like find out. I feel like that's going to be like a really big experiment or study.
Yeah, I think that would be someone else's postdoc.
Yeah.
But it's definitely the case that the Aroni lab is following this up and trying to figure out what role acyl sugars are playing in this like little ecological niche.
Okay. Do you have any more questions from the audience? Yeah, if that is not the case, I would like to thank everybody for attending again, and Jaynee for presenting.